Harshvardhan ji. Uh, actually, behind the scene, Dr. Viney Jain played a very important role, and he is working in this field from last almost 20 years or so. So he will just make a, presentation about this thing. Actually, now I will just hand over this responsibility to Professor Naresh Kumar ji, who was professor of biochemistry actually in the Baroda University, and he was also acting as Dean of Life Sciences Department in Nirma University. So I now request Sri Naresh Kumar ji to introduce Dr. Viney Jain and conduct rest of the session. Dr. Naresh Kumar ji. Kindly unmute yourself.
Good evening to everyone for this webinar. I'm pleased to introduce Dr. Viney Jain. Before that, I would like to state some simple things about glucose. Glucose, as everyone knows, is the energy required for most of the body. And generally what happens is glucose in the presence of oxygen, it makes a lot of energy. In the absence of oxygen, it makes the lesser amount of energy. And some 50 years back, scientists have found out some of the cancerous cells, even in the presence of oxygen, they don't produce high amount of energy, but they utilize very high amount of glucose. That was known as Warburg effect. That has become a very interesting discovery, and people were looking at how we can use this high glucose utilization by cancerous cells, and can we use that to specifically kill the cancerous cells. A lot of work is going on, and so one of the strategies is many of this metabolism is done by enzymes. So you use some similar molecules like glucose thing, and then try to mimic so that the pathway can be blocked. So this is the strategy which has been used, and 2-deoxyglucose is one of the similar analog, which is helpful in blocking the pathway. And people have been exploring its potential for cancer cell killing by using this molecule. And similar types of analogs are also used, the fluoroanalogs, deoxyfluoroanalogs, for monitoring the glucose utilization in our body, various cells, and even in the brain, that you're using by some specialized techniques, positron emission tomography, and so on and so forth. So this become a very, very interesting area. And the Dr. Viney Jain has been working on this area for a very long time. And he has been a biophysicist and a radiation biologist. And he received his postgraduate and postdoctoral training from University of Göttingen, Kiel, and Frankfurt, Germany. He has been the recipient of DAD German Academic Fellowship, as well as Alexander von Humboldt Foundation Fellowship for PhD program. This is a very prestigious fellowship. And he has been very active and published about 150 publications. And he's worked in the Physical Research Laboratory, Ahmedabad, All India Institute of Medical Sciences, New Delhi, National Institute of Mental Health and Neurosciences, Bangalore, Delhi University, and as a visiting professor and scientist at several universities. So he superannuated in 1998 from the post of director at Institute of Nuclear Medicine and Allied Sciences, New Delhi. Subsequently, he served as Professor Emeritus at B.R. Ambedkar Center of Biomedical Research, University of Delhi, and as a visiting scientist at Wright State University, Ohio, USA. Presently, Professor Jain is Professor Emeritus at the Jain Vishwa Bharati Institute, so the deemed university, Ladnan, and advisor to International School for Jain Studies in New Delhi. Professor Viney Jain is known for his original research contribution in developing differential modulations of radiation responses and their applications in improving cancer therapy. And he has been using this deoxyglucose as an inhibitor of glucose transport and glycolysis. So, and he has been worked with multi-center clinical trials on human patients with malignant brain tumors. What is interesting is Professor Viney Jain has worked in 1998 and now in 2021, we are seeing its application in the COVID viral infected cases. And this is also very, very unusual scenario where in the lungs where you have a lot of oxygen, lot of glucose is utilized, still this 2-deoxyglucose is making wonders. So we would like to listen to the intricacies of this phenomena from the main person who has worked for a long time and he knows each and every details. So we would welcome Dr. Viney Jain to give his personal discovery and his impressions of the current applications in the COVID treatment. Sir, we welcome you once again. Thank you.
Om Abraham Namaha. I सादर जय जिनेन्द्र सब श्रोताओं को और मैं बहुत खुश हूँ कि ये मेरे को अवसर मिला और ये अवसर में डॉक्टर कोठारी's इंस्टीट्यूट in memory of Kothari Institute and PRL scientists who founded the Jain Academy of Scholars, JS, are involved, and I'm very grateful to them, especially because I've been having personal discussions with Dr. Kothari, who was living close to INMAS campus, and I had several opportunities to visit him and have discussions on the quantum physics and Jain philosophy. Dr. Kothari has been, as you know, chairman of the University Grant Commission for several years, and also scientific advisor to the Defense Ministry. So he was instrumental in establishing also INMAS, that is Institute of Nuclear Medicine and Allied Sciences. And I'm also very happy to see that Dr. Bhandari, who was working in Physical Research Laboratory, has been the person behind the Jain Academy of Scholars, because my scientific career was a professional career, started from Physical Research Laboratory, where I joined in 1962 after getting my training in nuclear physics at the University of Kiel under Professor Eric Bagge, who was the student of Professor Heisenberg and was involved in the development of atomic bomb in Germany. He has written a book describing all the developments and his involvement. So Dr. Bagge, he was working on the time variation studies of cosmic rays. So after I finished my degree working with him, I joined Physical Research Laboratory, where Vikram Sarabhai was the chairman of the ISRO, and Professor Ramanathan was the director at that time. So Professor Sarabhai asked me to develop a scintillation counter telescope for the time variation studies. Now these studies, this involvement of my studies in the cosmic ray variations, and then trying to derive some understanding of the intergalactic space and the sunspot activity, that helped me to understand the variations, individual variability in the response of patients to drugs or to treatment. So I'm very happy that both these institutes have been involved in sponsoring and inviting me for this lecture.
Unmute करिए, unmute.
Who has given my introduction and for his kind words. Can I share the screen, please? Okay, now coming to the topic of my talk, developing unconventional, innovative, simple and cost-effective strategies to manage coronavirus pandemic, and the role of 2-deoxyglucose and methylene blue, I will touch upon it a little bit. So, the objective of this work is to, as described in this title, we want to develop simple and cost-effective strategies which can be used by a large number of people throughout the globe, throughout the world. Next slide. So I have divided my talk in these different headings. First of all, I'll provide a brief introduction and the history of 2DG research in India. Then development of 2DG for COVID, spelling galat, COVID-19 pandemic, and then what are the questions which remain and how we can have future directions for research to optimize the therapeutic efficacy, and what are the major implications of this work. Next. So the history goes back to very long time, because first of all, 2-deoxyglucose was used in USA in the East, and then finally to treat cancer patients in the NIH, National Institute of Cancer. There they used 2-deoxyglucose as intravenous injection, and it was to be given continuously for a long time. Now this continuous use of 2-deoxyglucose resulted in inhibiting the growth of the tumor, but the toxic effects were intolerable. And therefore, this clinical trial by Landau and his colleagues in 1958 was stopped, and this use of 2-deoxyglucose for cancer suffered a setback. Now, in 1970, I submitted my PhD thesis on the relationship between energy metabolism and growth in yeast cells. And there, after that, I developed the concept of using 2-deoxyglucose in combination with radiation at the Institute of Biophysics of the Goethe University, Frankfurt, Germany. And after coming back to and joining AMS as Assistant Professor of Biophysics, I continued this work within the framework of an Indo-German collaboration agreement between the two governments. And this was funded by ICMR, GSF, DARD, Alexander von Humboldt Foundation. In 82, I shifted to Department of Biophysics and NIMHANS in Bangalore as head of the department. And I worked there for about 5 years. And then I got an invitation from Dr. Arunachalam, who was at the time scientific advisor to Defense Ministry, to join there as director, because Brigadier Lakshmipati, who was then the director, was retiring. And Mr. Nagratnam, he recommended my name to him. So after long delay of more than 6 months, I decided to accept the invitation, and I joined in DRDO in 1987. And I was on deputation from NIMHANS for 3 years. So this is how the whole work of 2-deoxyglucose continued in different institutes and under the Indo-German collaboration. Next. So the work on Indo-German collaboration was summarized in a report written by me and Professor Polit, who was my guide of PhD and with whom I worked as a research associate after a PhD in Institute of Biophysics of the Goethe University in Frankfurt. This report was published in 1986 and summarized the work which has been done till then on model systems. Then in 1990, another volume on recent advances in radiation oncology was published. And in 1989, we organized the international symposium, Indo-German symposium on this topic, where experts on radiation physics, biology, and oncology, they joined from both from India and from Germany. And this conference gave an impetus to the interest of 2-deoxyglucose in oncology. So I will briefly describe a little bit about the basic and preclinical research and development of appropriate model systems. And the importance of model systems is very important because you have to try to simulate the conditions in the tumor cells, or in this case now in the COVID-19 patients. And for this, I think the simple models are very useful to understand the basic mechanisms. And that's what we use, yeast and its mutants. Now, the simple model systems are really very much required to understand mechanisms, just like in physics, hydrogen atom was used for the main discoveries in atomic physics and quantum biology. So after that, we systematically developed, worked more on the in vitro models and also in vivo models involving normal and tumor-bearing mice. And then also I would like to emphasize that importance of the theoretical models, theoretical studies, because some of these work which we have done have given very important information. And among these in vitro models, I'll like to point out two things. One is the development of perfused agarose threads, which we developed because this perfused agarose thread system allows us to simulate the conditions and the microenvironments of the tumor cells. And one can study this using the MRI spectroscopy, which we used in first of all in the Tata Memorial Center, in the Tata Institute of Fundamental Research, where Professor Govil was the head of the national facility of NMR work. And he gave us some time on his facility, but that was not enough because there was a lot of pressure of other studies. Then organ cultures from the human cells, normal and the tumor cells. This was developed by at AIMS, first of all in skin cultures and then also in brain tumor in the NIMHANS. And Dr. T.N. Chapekar, who was the assistant professor in Department of Biochemistry at that time, and he was an expert in this. So he introduced us and taught us how these organ cultures can be used and developed. Next. Now, I'll spend a few lines on the use of Saccharomyces cerevisiae yeast to study basic mechanism. What are the advantages? First of all, it's a single eukaryotic cell system. That means it has a nucleus and also a nucleus which contains the DNA. And it has a homology to human genes, and number of mutants with energy and nutrient deficiency are available. Easy to manipulate the microenvironment, metabolic and growth states, and can be grown easily in the batch or continuous culture in different media and environments. So it has several advantages, and that's why we concentrated our work first of all on yeast. Now, what are the significant findings of our contributions in this field? So as first of all, the work on yeast cells shows that 2-deoxyglucose inhibits the energy-dependent cellular repair processes. And this proof of principle was shown then in malignant cells in vitro as well as in clinical trials in vivo. So we developed the idea that 2DG works as this radiosensitizer, differential radiosensitizer in tumor cells, and it needs to be given only for a short time in combination with radiochemotherapy of cancer. And therefore, the toxicity which was associated with the continuous administration of high doses of 2-deoxyglucose observed in the National Institute of Health in Bethesda, that could be avoided. And therefore, there was a renewed interest in 2-deoxyglucose work and its derivatives, as Professor Naresh has indicated in his remarks. Second thing is that 2-deoxyglucose enhances error-free repair in normal cells. And 2-deoxyglucose may inhibit recruitment of tumor stem cells. Now, this is the contribution of theoretical models. Professor Kamlesh Bhaumik, who was the assistant professor in biophysics department at AIIMS and then transferred to NIMHANS, he developed a theoretical model, and that model predicted that 2-deoxyglucose will inhibit also recruitment of tumor stem cells, because only the inhibition of repair was not sufficient to explain the experimental observations. This was later on experimentally verified in the mammalian cells and in tumors in various laboratories throughout the world. Then 2-deoxyglucose also inhibits the migration of malignant cells. This work was done in collaboration with the Tata Memorial Center Cancer Research Institute, where I was deputed for a couple of months, 3 months, to carry out certain experiments. And in collaboration with Dr. Gothaskar, we showed that the migration of malignant cells may also be inhibited. So eventually, all this work showed, however, that effects of 2-deoxyglucose are heterogeneous, and that depends on the metabolic state of the system and on the 2-deoxyglucose ratios achievable. Because some of the tumor cells, which were not so much dependent on glycolysis, they had much lesser effect. And as it was mentioned by Dr. Naresh about the Warburg effect, now Warburg effect was discovered or published in 1924 by Dr. Warburg in Germany, but later on there was lots of skepticism, and the interest in Warburg effect research was reduced. But now in the last 2 decades or so, the research on Warburg effect has increased a lot, and interest in this work has increased because of the use of FTG 18F DG with the PET, showing that most of the tumors, especially those which are resistant, they showed a high uptake of FTG. Next. Now, let me come to the history, how I developed this concept. Now, the coupling between the energy yielding processes and energy consuming processes in living system is tightly coupled, and this is very much evolved because the living systems use this regulation between demand and supply very wisely. But sometimes this balance between the energy yielding processes and energy consuming processes gets disturbed, because some of the examples are, for example, tumor growth, virus replication, and some of the other neurodegenerative diseases, thyroid disorders, and so on, epilepsy. So it has implications in many, many disease states. And we will confine ourselves in this mainly on the tumor and the virus replication. Now, the energy supply in the living system is through the formation of adenosine triphosphate called ATP, and this is the major pathway. And the ATP can be generated by glycolysis or the respiratory pathway. The glycolysis pathway produces only 2 molecules of ATP per molecule of glucose, whereas the oxidative phosphorylation in advantage of glycolysis is that it produces the ATP in a very fast manner. So whenever there is a fast demand of energy, then preferentially glycolysis is used, and this is called the Warburg effect. But the quantitative and continuous measurement of cellular ATP flux is challenging, is very difficult. Most of the people determine only the levels, but the levels, as we have learned, is not very closely correlated with some of the processes, ATP-dependent processes. It is the flow of energy through ATP which is important. And therefore, if we can monitor ATP dynamics, it can predict many disease states and determine the efficacy of drug treatments, and it could be used to modulate or actuate the therapeutic molecular release in response to cellular energetics. So it is important. Now, just to elaborate this is a paper I wrote in 1970 and is from my PhD thesis work. Pointer. So after the glucose transport in the cells, you get glucose 6-phosphate, and this has then branches to many different pathways. But what is important is that the energy is also stored in glycogen or cellulose, so the excess energy is stored. And similarly, this is the respiration oxidative phosphorylation in mitochondria. Here also, the lipids and the fat, they can store the energy, and that can be done again, brought back whenever the use is necessary. And this ATP produced by both the pathways is then closely linked with the function of the cells and growth, or in this case, and also in various other processes. Next. So now I come to the dependence of repair process on the ATP flux, and this was measured in yeast in the respiratory deficient mutants, because here the rate of glycolysis is then proportional to the rate of ATP production. And important finding is that this dependence is a sort of sigmoid curve. That means there is a threshold, and there is then the rapid rise, and there is then saturation. And important thing is that the repair depends on the type of damage which is induced. For example, with X-rays or UV light, which produce mainly the dimers, and the sorrelin induced damage in the presence of light called PUA, and here only some cross-links in the DNA are produced. And with X-rays, different kinds of damages are produced: base damage, or single strand breaks, double strand breaks, and some clusters of these lesions. Next. Now this we also tried to see how it happens in the brain. And brain can be, there are several methods which can give some clue to this. One is the use of PET using analog of glucose, or the oxygen supply, some radioactive isotopes using oxygen. And here, but we have used the Bolt signal change, which measures the blood flow and the oxygen utilization. And we developed a method of finger tapping rate. The finger tapping rate utilizes the energy, and this method was developed by Dr. Subhash Khushu, who worked for his PhD with me at INMAS. And he was head of the NMR Center. So as you see here, there is a linear, again, linear, see, there is a linear dependence, and then comes the saturation, and with very low, there may not be much effect. And this can be seen also in the images obtained by MRI. Next. Now, a very important fact which we should remember is the heterogeneity of microenvironments and metabolic state of cell population in tumors. Now, this is because the rapidly growing tumors, they are having insufficiency of blood supply, because the angiogenesis resulted, it cannot be sufficient, and therefore there are the gradients of oxygen availability in the tumor. That's cells which are remote from the blood-carrying arteries or veins are hypoxic, completely hypoxic. And then cells which are nearer to the blood supply vessels, they are more or less oxygen sufficient. So the imbalance of the demand and supply in energy and nutrients creates heterogeneity in metabolic and proliferation status of the cells. And similar is this case in case of COVID-19 disease. And as the disease progresses, the respiration becomes difficult, and there may be then hypoxic cells which are being developed. And the COVID or virus infected cells, they also need a lot of energy because of the rapid replication of the virus in the host cells. Next. So this is the rationale why we try to use 2DG in the COVID-19. Now this is again trying to explain how 2-deoxyglucose works in normal and cancer or virus infected cells. As you see here, there's more of glucose transport, 2DG transported inside the cells because it's being an analog. It uses the same system of transport inside the cell, and therefore reduces the glucose transport. But formation of 2-deoxyglucose 6-phosphate once inside the cell by the hexokinase, that does not proceeds further, and it blocks the pentose phosphate pathway as well as the glycosylation pathway. Next. So again, this summarizes this. The 2-deoxyglucose phosphate accumulation or retention or trapping in these cells that inhibits the glycolytic pathway, HMP pathway, and alters glycosylation, thereby changing structure of glycoproteins, very important for the virus. So this is a very important pathway because the 2-deoxyglucose replaces glucose, and that changes the cell surface glycoproteins and also the protein coat of the virus. Next. Now, just to give you some data, I won't bother you with lots of data because it's too much, but just to show that in these chromosome aberrations and survival of normal mice after the radiation, that actually reduces. This is the chromosomal aberrations, structural aberrations after 50 hours of post-irradiation, and this is the 30-day survival in percentage in the irradiated mice, whole body irradiated, and in the group which was given 2D. Because you see there is about 20% difference, which is statistically significant. Next. So important thing is that 2-deoxyglucose protects the normal cells. Now again, this is shown once in cell cultures of peripheral blood leukocytes and HeLa cells. In peripheral blood leukocytes, 2-deoxyglucose reduces the micronuclei frequency, and in the HeLa cells it increases the micronuclei frequency and causing cell death. Next. So from this, we developed a model that the DNA strand breaks, if repaired, may result in chromosomal breaks, and competitions between rates of process of error-free repair, error-prone repair, and fixation of damage determines the cell fate, whether the cells will die or survive or get mutated. Next slide, please. This is again shown in this simplified model. Now, here, the energy stress also induces endoplasmic reticulum stress, and this is responsible for the apoptotic cell death. So these are the different types of cell death: necrosis, apoptosis, autophagy, and immune system mediated. And the spectrum of the death, cell death types, that changes with the metabolic state and with the 2-deoxyglucose administration. So this has important consequences because, next one, because the immune system responses to cell death, they trigger inflammatory responses by releasing danger signals which can promote either generation of immune responses to antigens in and around the dying cells, which are useful for eliminating the infection, but they can also cause tissue damage, contributing to the pathogenesis of a number of diseases. And so this is a double-sided sword, which can be useful or which can be also harmful, and the type of cell death influences the outcome. Next slide. The mechanisms involved in this are very, very complicated, and a lot remains to be done. So the important point here is the distinction between the self and non-self in the immune system. And this distinction between self and non-self is very important and plays a big role in the in all aspects of life, also in our behavior, reflected also in our behavior. Now, as I mentioned, there is also the highly specific T and B cell recruitment from more distal sites, and this leads to a rapid but indiscriminate response. And this unregulated overreaction may give rise to cytokine storm. This is important for the COVID-19 tackling. And as I mentioned, the understanding of these complex mechanisms and the interactions and the interdependence of the various pathways is still very deficient, and more research work will probably give us a better understanding. And the COVID-19 patients, and if one studies this, will perhaps lead to better understanding. So now coming from the east to the cancer patients and to the COVID-19 patients, that's a long journey. And I'll briefly mention about the clinical trial on thyroid carcinoma patients. This was carried out in AIMS when, with the Department of Nuclear Medicine. Professor A.K. Basu was then the head. He treated 5 patients, but then it stopped because the non-availability of 2-deoxyglucose. We had to buy it from Sigma, and that was very expensive, and therefore could not be we could not afford it, and therefore this clinical trial on carcinoma patients they stopped. Then, as I indicated, clinical phase 1, 2, and 3 trials on brain tumors in glioblastoma patients, they were started in NIMHANS in 1986 with the collaboration of Kidwai Institute of Radiation Oncology. The first patient was treated by Professor B.K. Mohanty, and actually he told me that he could not sleep the whole night after giving 2-deoxyglucose, was always with the patient. But then I shifted to INMAS and I continued in this work, as I mentioned, with after the Indo-German Symposium. It's got a little encouragement, and a number of institutes in India participated in this phase 1, 2, and 3 trial on brain tumor patients. And this continued up to 2006. But then this work again is stopped because though the patent was transferred from DRDO to Dr. Reddy's lab, but somehow or the other, they did not manufacture it and supply it, perhaps because of economic reasons or whatever. Now, when COVID-19 pandemic came, then I got the idea that why can't we try it on COVID-19 patients? Now, in 1970s in USA, there were a number of in vitro studies on the virus-infected cells and use of 2-deoxyglucose, and they had shown that 2-deoxyglucose can be used in viral infections. Actually, a patent for use in herpes virus infections was obtained in the USA. But again, I don't know the reasons, but the work did not begin there. And then in India, when I was aware of those literature, and I suggested to Dr. Rakesh Sharma, who was then, after retirement from DRDO, as director of Food Research Institute in Mysore. He joined as the vice chancellor of Savita University in Chennai. And so I phoned him and suggested that he should start this work of clinical trials in his university. But he expressed his inability because he said they don't have any facility for this at that moment. But he said that we can do some theoretical modeling on this because his students, Dr. Pallavi Thakur, he had joined Patanjali Research Institute research group, and he said that she can do this theoretical modeling together with him. And so he contacted her, and Dr. Pallavi Thakur got the permission from the Patanjali Research Institute, and she carried out this work, which was then submitted for publication in April 2020, and is now accepted and will appear very soon in Annals of Academy of Medical Science. So then I also contacted INMAS director at that time, Dr. Tarun Shekhri, and suggested to him that he should start this COVID-19 patients clinical trials. He contacted then his experts, that is Dr. Sudhir Chand and Dr. Anant Narayan Bhatt, who had been working on this. And after their discussion, Dr. Shekhri was convinced, and he tried to then get the permission of the ICMR and the drug controller of Government of India. And after lots of discussion, this sanction was given for phase 1. Then they presented the data. Phase 1 got permission for second phase and in third phase. Next. Now I just want to show this slide that the dynamics of blood glucose and 2DG levels as we measured in brain tumor patients. Now, important thing is that the glucose levels in the blood, they increase after 2DG administration. So there is hyperglycemia in these patients, but they show symptoms of hypoglycemia. And all the 2-deoxyglucose levels are shown here. So it remains high only for a very small time. And what is important, as we learned from our in vitro and in vivo model system work, that the molar concentration ratio is important for determining the responses to 2DG. And this ratio is really very small, only for first few hours, couple of hours, and then it declines rapidly. So this is important and must be taken into account. Now, we come to this COVID-19 infection and what are the important steps. As most of you are aware, the first step, that is the spike glycoproteins, they are binding to the angiotensin converting enzyme 2, AC2, on the receptors on the cell surface. And this then helps in the entering of the virus to the host cells. And so this is a very important target for developing the effective treatment strategies. And most of the drugs which are now becoming available, or even the vaccines, they work on this step. After entry, the virus makes use of the host cell machinery to rapidly replicate itself. And as we have shown, and as is well known, that increased glucose transport and glycolysis, just like the tumor is increased. And an important thing also to see that the immunological response resulting in cytokine storm or acute respiratory distress syndrome, ARDS, and secondary infections. These complicate the patient survival after the COVID-19 infection. That's in the severe patients. And another problem is that the virus goes on mutating, and that changes then the infectivity and the symptoms and the time course of the disease. So an important thing again is that the time course of COVID-19 disease is very heterogeneous and dynamic, keeps on changing with time. And from asymptomatic to mild to moderate and severe. Next slide. So this is just a case report showing that 2-deoxyglucose is really increasing the SpO2 and maintains SpO2 at more than 90%. And this is a report sent to me by Dr. Viney Jain, who is a medical consultant at IFCO. Next slide. But the work in clinical trial, phase 3, conducted by DRDO, especially Dr. Anant Bhatt and Dr. Sudhir Chandna, and the DRDO team, that has thrown some light on the safety and efficacy of 2DG. Now, phase 3 clinical trial was divided in 2 parts. And in the phase 3, finally, 45 milligram per kilogram body weight of 2-deoxyglucose dissolved in water was given in morning and evening. And this was given mainly to the moderate and severe COVID-19 patients who required oxygen support in the hospital. And to assess the safety and efficacy, WHO 10-point scale was used. And this on the endpoints which have been measured till now are requirement of oxygen and hospital stay. The data on mortality, on the survival of patients and the quality of life, that remains yet to be determined. The data needs to be analyzed and on a large number of patients. This will be done in due course of time. But the present scenario is that the statistically significant reduction of more than 1 point was observed in 2-deoxyglucose plus standard of care as compared to standard of care control group. And no significant adverse events were reported in patients given 2-deoxyglucose. So these are the 2 results which we can rely on, and this opens the gate for wider emergency use of 2-deoxyglucose, for which the permission was given on 17th May And now the Dr. Reddy's lab will provide 2-deoxyglucose to hospitals, government hospitals, and some of the private hospitals. And later on, we hope in a couple of weeks, the supply can be also given to chemists who supply the drug. So this is what the present scenario is. Now this is lots of questions have been raised about this, about those, about the mechanisms, as well as about the permission given by DCGI, and some people say it is a premature permission, and without any published reports and without doing clinical trials on a large number of patients. But then I feel that this is justified, because you have to make the decision based on the benefit and risk ratio. And in an emergency situation like COVID-19, this there is a lot of benefits and less risk, I feel. And therefore, my personal view is that it is justified and appropriate under the present situations. So now, how 2DJ may work as an antiviral agent, I've already explained, but once again, the in silico modeling suggests that 2-deoxyglucose can inhibit the binding of the spike protein with the ACE receptor. Now, if this is verified in experiments, in vitro experiments and in vivo, then it has tremendous implications, because then 2-deoxyglucose, which is given orally in a very simple method, and this can be used on a wide scale, even to in the asymptotic or the mild conditions, and also in rural areas. Next. Well, this I've already explained. Now I come to methods by which we can increase the therapeutic efficacy, because as I mentioned, there is a large variability. Some patients have shown immediate and very good response, even in the severe patients, but others have shown no effect. Therefore, we have to further carry out research to develop approaches which are cost-effective and feasible on the large scale. Now, there are a number of approaches which are being tried all over the world, but I will discuss here only combination with methylene blue photodynamic therapy, which is a very cheap and cost-effective alternative. Now, methylene blue is known for a long time, and it has been used as a dye in textile industry and as a stain in histopathology. And several medical applications in earlier days in the 19th century, they include application as an anti-malarial drug and methemoglobinemia and cyanide poisoning, where oxygen supply is affected because the methemoglobinemia is not able to carry enough oxygen, and also after cyanide poisoning, the supply of oxygen is very severely affected. So this can be then used certainly in the enveloped viruses, including the SARS-CoV-2. And this has been shown in vitro results way back in 1970s in USA. And this is also methylene blue is included in the WHO list of essential medicines because of these medical applications. Now, PDT involves the uptake of a photosensitizer followed by radiation with visible light of an appropriate wavelength, which depends upon the absorption spectrum of the photosensitizer used. And these photochemical reactions, they trigger generation of reactive oxygen species, mainly singlet oxygen, leading to the damage of biomolecules and organelles. Now, this has several advantages compared to surgery, chemotherapy, and radiotherapy. First of all, there is no long-term morbidity. And the use of MB PDT does not compromise other treatment options. And most importantly, it's inexpensive and available in the market as approved drug. So this can be immediately tried. This is just the absorption spectrum. You see most of the light is absorbed in the red region above 600. This shows how the singlet oxygen generation after photodynamic therapy can damage the RNA or DNA. And now we have some experience with methylene blue steam inhalation. This was being used. This is being used in Maharashtra, Madhya Pradesh, and Gujarat. Number of doctors are using it. And what are our experiences? Dr. Viney Jain has tried it on 10 patients of COVID-19, and she has found very good results, very effective results, and the drug is improving the oxygenation of blood. That should be a trial. Similarly, homeopathic physician Dr. Poonam Jain has tried this on post-COVID complications, and she has also found very
screenshot. Screenshot.
Okay, I think my time is up. So, what are the conclusions? So, this use of methylene blue alone or in combination with visible light irradiation should be investigated further. Combination of 2DG with methylene blue can, in the dark or with PDT, can improve the efficacy of therapy and lead to reduction in the pain and suffering. And that is the main objective of the biomedical scientists. And I think this objective can be met with trying this. So we should encourage multidisciplinary research, which will lead to this development of non-conventional, simple, and cost-effective treatment modalities, which will certainly be able to be used also in rural settings and where most of our population is suffering. Next slide. In the end, I will like to acknowledge the funding agencies in Germany and various institutions, that is ICMR, DRDO, Department of Atomic Energy in India, in Germany, the Society for Radiation and Environmental Research called GSF, German Academic Exchange Service, DRD, and Alexandra von Humboldt Foundation. And institutions mainly Goethe University, Frankfurt, AIMS, New Delhi, DIMANS, Bangalore, INMAS, Delhi, where the clinical trials were conducted in INMAS. And this is the very important role played by Dr. Anant Bhatt and Dr. Sudhir Chandna, together with the DRL scientist. And then University of Essen, Germany, Patanjali Research Institute for the theoretical work showing that the 2-deoxyglucose may inhibit the entry of the virus, of the Savita University, Chennai, and JVBI, Ladnu, where I'm working now. So, next. Now, some of the mentors and teachers and my students, which have played a very great role, and I have been fortunate to have very capable and teachers, and also my students, very, very efficient. And several colleagues whose names are mentioned here, but there so many participants, not all of them have been mentioned. I beg to be forgiven if I've omitted some. Next. And last but not least, I have to express my gratitude to my family, my father, Sri Aradas Jain, my mother, Srimati Kanta Jain. My father, he supported and encouraged me, and he took loan to send me to Germany. And when I resigned from PRL and joined as a research fellow of ICMR in the AIMS, he never objected. He agreed with me. And also my mother to give me all the sanskar of the Jain lifestyle. See, I remember very clearly as a child when I was trying to kill an ant going on the floor, my mother scolded me and says, "Why are you doing it? उसने तुम्हारा क्या बिगाड़ा है? उसको क्यों मार रहे हो?" And therefore, I learned the significance of non-aggressiveness and ahimsa. And my wife, late wife, Asha Jain, she took all the responsibility of educating my children, Priyanka and Anurag, and also all my social obligations, and gave me free time to devote to my research work. So my family, I'm very grateful to all of them. Otherwise, the work would not have been possible. So thanks to all my audience, and also to the organizers of this talk, Dr. Bhandari and Dr. Pokharna of the. So with this, I would like to end my talk, and I will be happy to answer any questions or queries to the best of my knowledge. Thank you very much. So.
Sir, I am Professor G.C. Jain. I have two simple questions to ask. One is that I am a veterinary scientist from Indian Council of Agricultural Research, and I also worked at National Dairy Research Institute, AIMS, and Houston, the Health Science Center, and California. You have rightly said that for the development of cancerous cell, we require a hypoxic environment. my submission is that besides hypoxic environment, we also require acidic environment, and that too with the heavy load of free radicals. The second question is that we talk of mutation. When we see the microstructure of the corona molecule virus, there is a very short DNA molecule in it. However, the RNA molecule is very prominent, and that's why I wonder how mutation takes place without the long chain of DNA.
Well, regarding the first question, as I mentioned, the environment in the tumor is very heterogeneous.
Yeah, that's right, sir.
That depends on the metabolic state, and
Right.
our work on model systems have tumor-bearing mice had shown that the cell loss from the hypoxic fraction is much more than
Much more.
the eoxic cell population of the tumor.
Exactly.
It was done by a very ingenious technique of double labeling developed by Professor Feinendagen of the German Nuclear Center, Institute of Nuclear Medicine,
Yeah,
where
that's true.
I spent some time to carry out these experiments. So
Right, sir.
almost all the tumor cell populations will be affected, but the effect will be more in hypoxic fraction.
Right,
Because
sir.
of the Warburg effect. So you are right there. And regarding the second question, the 2-deoxyglucose has been shown to interact with the glycoprotein, a spike glycoprotein, which facilitates the entry of the virus inside the cell. And therefore, the replication of the RNA, which requires a lot of energy.
Sir, this is true. This is true. My question was for DNA, because mutation is nothing but a change of gene. When the DNA is very weak in this coronavirus 19, so how, we are talking nowadays of variant mutation, variant viruses, all those things. So, and why it is so frequent of talk of this mutation when the DNA sequence is very, very, very weak?
You see, the DNA will also lead to mutation, and RNA changes also will lead to mutation. Important thing is
leads to mutation, but the RNA, it is indirectly way of this is through messenger. Only it helps in copying, the digital copying, not the mutation. DNA is responsible for mutation when there is a change in gene.
The replication of all the elements of the virus requires a lot of energy, and that is inhibited. Second thing, as our theoretical model shows, that the entry of the virus inside the host cell will be also inhibited. That is interaction between the glycoprotein coat of the virus and the cell surface. Please let me finish.
Okay.
And therefore, one important implication is that 2-deoxyglucose is not specific for COVID-19. It is a broad spectrum antiviral agent, and it will inhibit many other viral infections. For example, in the hospital, there is a hospital infection, and several other diseases which are caused by viruses will also be prevented and cured by 2-deoxyglucose. So that is the point with very important implications.
Shall we move on to other questions? There are many things.
Okay, sir. Thank you so much, sir.
Yeah. So now we have another question from Sri Navin Mehta. He is asking, why were the trial done only on thyroid cancer and brain cancer cells? But breast cancer, prostate cancer are the most common cancers. and research has to be done on the effectiveness of this treatment on most prevalent cancers.
Quite right. The thyroid cancer was undertaken because in AIMS in nuclear medicine department, thyroid cancer was being treated by radioactive iodine. And therefore, these patients were easily available. And therefore, we started with those. And secondly, about the prostate and breast cancer, some of the clinical trials have been initiated in after 210 in USA, and they have also shown some benefit. But the main point is that the availability of 2-deoxyglucose in the commercial market is not there, and they all purchase from Sigma, which is very expensive. So the availability of 2-deoxyglucose has inhibited the application of 2-deoxyglucose in cancer patients and also in COVID-19.
So does it mean that now we are able to produce so much of 2-deoxyglucose can now be tried for other tumors also?
Exactly. This work will be undertaken again by AIMS and Dr. Dinesh Singh, who told me on telephonic conversations that they are very much interested. But then this all depends on how cheap and how frequently, how much can be produced by Dr. Reddy's lab. And therefore, suggestions have been made that this technology should also be transferred to other manufacturing firms. And once it is becoming available, then I think work will pick up.
Okay. Now we have one more question from Sri Narendra Bandari saying, excellent insight into the process. My appreciation. A simple clarification. You mentioned autophagy. So in cancer, as well as in COVID, fasting or stopping intake of sugar should help.
Yes, autophagy, role of autophagy is very important, but as I mentioned, the immunological response depends on the spectrum of the cell death type. And 2-deoxyglucose is increasing the necrotic cell death, apoptotic cell death, and also autophagy. But there is a competition between these, and the exact role depends on the spectrum and how much you are giving the 2-deoxyglucose and for how long, because the prolonged use of stress due to starvation then promotes autophagy. But the autophagy certainly plays a role, but its role needs to be further investigated. This is my view.
Okay, Professor Shamlal was also asking the similar thing. Do we know about the mechanism of 2DG on autophagy?
Yes, autophagy has been shown to be induced by 2-deoxyglucose, and in vitro studies have shown in several countries. We have not done any of these experiments, but in USA and in Germany, this work has been carried out.
So, Sri Madan Gobole is asking, is there any difference between autophagy and autophagy flux?
No. And I didn't hear autophagy
flux. I'm not asking that question. I'm saying there's a difference between autophagy and autophagy flux. Autophagy flux gives the real picture. Sir, my question is little different about the methylene blue. Are there any trials planned?
There are some trials going on in the USA and Europe.
In India.
That's
Sir,
what I
India,
learned.
Pune and Bombay, they are using it as a vapor, methylene blue vapor.
Yes, that's what I said. It is steam inhalation or nebulization. These are the two
Nebulizer.
methods by which it can be given, and the dose used is very small. So it is completely
small, right.
non-toxic. And I personally have been using it for more than 2 weeks, and I'm feeling fine.
But there should be a planned trial on this. That is what my.
Yeah, that's what my suggestion was, that immediately bigger clinical, systematic clinical trials must be started in India.
Yeah.
That's what my instruction.
Yes, sir.
Yeah.
Thank you, sir.
There are
Autophagy and apoptosis, there are differences. Apoptosis is a programmed cell death that is occurring in a natural way during the development of body, as well as maintaining the homeostasis in the adult mature body, because cell loss and cell death and cell recruitment from the stem cells, these are very normal processes going on in the healthy individuals. And therefore, autophagy is also important, because autophagy leads to the protection of cells during starvation, during stress. And if it is for long duration, then autophagy becomes important for recycling the energy and nutrient and because autophagy leads to the lysis of cells. Apoptosis does not usually.
there are general questions about phase 3 trial data and its publication. What is the status and when it is going to happen?
Yes, the data is being analyzed, because there has been a lot of delay, because the transfer of data to computers has been delayed, because the data entry operators, they did not come during the COVID-19. Now the data has been transferred, and then it will be completely analyzed, and the publications will result soon. Dr. Sudhir Chandna and Dr. Bhatt has assured me. And also Dr. Reddy's lab is in a hurry to publish this, but it has to be properly analyzed. So I think the publication will come as soon as possible.
Are there any side effects seen in the clinical trials?
No, that's what I mentioned, that no side effects, serious adverse events were observed in the 110 or more patients combined with phase 2 so far. And we also did not observe any side effects, serious side effects at a much higher dose given to the glioblastoma patients. And this work was published by Dr. Dinesh Singh and Professor A.K. Banerjee of AIIMS, and in the dose escalation work in 2005, this paper appeared. So I think as far as safety of 2-deoxyglucose is concerned, and given orally, there seem to be no serious side effects.
Do you think inhalation type of thing, nasal spray or something is going to be better?
Definitely.
Yeah, nasal spray is also very good method. But it's easy to take it as a steam inhalation.